I want to introduce you to electron carriers. God, what a mess this board is.
I know, right?
This electron carrier, are we filming?
Yes, sir.
This electron carrier up here is called NAD. And it's a molecule in your cells that's built to carry electrons. And it's actually, I don't know if you can recognize this, just cut it off right there. Does this look like anything you're familiar with? Just this part of it?
ADP.
Cholesterol.
A base?
Yeah,
Like a...
That's what it says.
No.
I haven't heard it yet.
It looks like a piece of DNA.
This is another one right here. Yeah, it looks like a piece of DNA. Who said that?
Okay, well,
Andrew?
Good job, Dylan.
Emmy.
But what is a piece of DNA called? I haven't heard that.
Nitrogen base.
I still haven't heard it.
Nucleic acid.
A
Strand.
piece of nucleic acid.
Okay, I said it.
I haven't heard it yet.
I said it.
You haven't heard it.
Nucleic acid. I'll
No,
scream
that's
next.
not a piece of nucleic
Guys,
acid. I'm talking
Chromosome.
about what's a piece of nucleic acid. What's that?
Gene?
No?
It's a cheese.
I just heard it. It's a nucleotide.
Oh.
A nucleotide. A piece of DNA. I don't know if I have it. Oh, here's a nucleotide right here. Got one. A nucleotide is a sugar, a phosphate, and a nitrogen base. That's what this is right here. There's the sugar, there's the phosphate, and there's the nitrogen base. NAD is two of them, two nucleotides hooked together. Can you see there's a nucleotide there and a nucleotide there? And what it'll do, this NAD, is it has a positive charge. I don't know if you can see that little positive charge there. See that little plus? And this is an electron acceptor. It's positively charged. It'll float around. I have a model of it right here. Here's what NAD looks like on my magnets. Let me turn the light on so you can see it a little better. There's what NAD looks like with my magnets. See, it's got two nucleotides. One's an adenine, and one's says N on it. That stands for nicotinamide. NAD stands for nicotinamide adenine diphosphate.
That's kind of a long word.
Nicotinamide adenine diphosphate. Can you say nicotinamide adenine diphosphate? Hard to say. That's why they just say NAD. If I say diphosphate, does that give you any clue about what's in it?
Phosphate.
Two phosphates. One there, one there.
Go Carter.
So you can see one on top and one on bottom. And so these will accept electrons. And so here's how this works. These are electrons, and the NAD will pick up, it's positively charged, it can pick up two electrons. If something has a single positive charge and it picks up two things that are negative, what kind of charge does that give it?
Negative.
A single negative charge. But it will also pick up at the same time a hydrogen ion, which is also called, do you know what a hydrogen ion is also called? Also known as?
I believe it's proton.
It's a proton.
Next one, Andrew.
Andrew's on the science bowl team, so he knows.
It's gonna be me.
A hydrogen ion is a proton. They're the same thing because a hydrogen atom has one proton and one electron going around it. That's a hydrogen atom, just a proton and electron. The electron goes around the proton, and that forms a hydrogen atom. If you take the electron away, you have a hydrogen ion. An ion is when you remove the electron from an atom or you add an electron to an atom. But in this case, we remove the electron, and now it's just the proton. It's a positively charged proton. That's a hydrogen ion. Same thing. So if the NAD, which is positively charged, picks up 2 electrons, it will also attract a positive hydrogen. And now it is a neutral atom that we call NADH. NAD becomes NADH. Whoops. So here's NAD. When it picks up electrons and a hydrogen, it becomes NADH. Can you see up there where the hydrogen it picked up is?
Yeah. The very top.
Yes, question.
So when did you say the hydrogens are positive?
Hydrogens are positive.
So
Hydrogen
you said
ions.
the NAD had a single negative charge, but you added 3 things that are positive,
Yeah.
and now it has a neutral. So do each one of those have like a half a charge, or how does that work?
So NAD has 1 positive charge, plus 1. If it takes on 2 electrons, which are each minus 1, what's the charge now?
Negative.
Negative what? Negative 1, negative 2, negative 3?
Negative
1.
Negative 1. And then it takes on 1 positive hydrogen, which is positive 1. What's the charge now?
So okay, yeah, neutral. Okay.
So NAD is an electron carrier, and you'll be learning about NAD a lot. There's another electron carrier, which we'll look at. It's called FAD. And FAD is a little bit different than NAD. It's a little bit bigger. It looks a little different, and it can actually hold 2 electrons. Whoops. And it can hold 2 hydrogens. And the cells use these electron carriers. We're specifically going to look at them in the mitochondrions. This is a big mitochondrion. Can you tell?
Yes.
We're going to learn all about what happens inside and outside the mitochondrion in this process of what we call aerobic respiration, which is when you take glucose right here and you break it down, you break it apart, and you get the energy out of all these glucose bonds. Every little line you see there is a bond that stores energy. And the cell, the mitochondrion, has a way of breaking those bonds and releasing the energy and using it to make ATP. Now, that's what the next slide's about. It's talking about ATP again. We already looked at ATP, didn't we?
Mm-hmm.
So, ATP forms from ADP and P coming together, click, to make ATP. The mitochondrion can do this. It has a way it's evolved of moving electrons, of breaking down glucose. The NADHs and the FADHs carry the electrons that came from the glucose molecule to this thing right here. It's called an electron transport chain. And the electron transport chain pumps hydrogen through. Pumps hydrogen through this way, and the hydrogen comes back through an ATP synthase molecule, and we end up making ATP. Do you guys, how much of this did we do in regular bio with y'all? I get confused.
Yeah, we did a long
We
bit
touched
about
on
the-
it, but I think we
We
touched
talked
on
about all the
We
like
talked about the
different-
electron chain and,
Yeah, and
and
the
the
glycolysis.
Okay, so some of these words are familiar. You probably don't understand the whole process yet, but we have like 5 days to spend on this. And by the time this is over, every one of you is going to have to model this for me, exactly how it happens, and then you'll know it, and then you'll do well on the test. So, is this from this same section?
Yes,
Yes, sir.
it is.
So ATP, this ATP molecule often phosphorylates other molecules. Phosphorylation is when the ATP molecule drops off its phosphate somewhere.
What's a fancy word?
And so here's an ATP molecule coming in. That's a protein. Can you tell that that's a protein?
Yeah.
Look, it's got a little alpha helix there. Huh? It's got a little beta sheet right there. That's a little beta sheet. This is a single chain. This is a long primary structure that's folded into a tertiary structure. Anyway, that protein gets phosphorylated by ATP. ATP turns into a DP and drops off a phosphate. Now this protein is phosphorylated. The phosphorylation of a protein will often change its shape. If you add a phosphate group to this chain, what do I do my protein chain? Drop it off over here. Aha! Maybe when I lose my protein chain. So here's my protein chain. It's all folded up into whatever shape we need. If you phosphorylate it, see, ATP is a very common molecule in cells. There's a bunch of it floating around. And sometimes, under the right conditions, maybe it attaches to an enzyme. The active site has a protein in one part and the ATP in another part, and then the P gets transferred to the protein. And the ADP goes away, and now you got this phosphate connected to the protein. Well, that'll usually change its shape. So phosphorylating proteins is a way to change the shape of a protein. And then when the phosphate group comes off, it'll go back to the correct shape. So this is often how cells change shapes of proteins to get something done. Do you remember when we studied the cell membrane? There were proteins in the membrane. Did we study this already?
Yeah.
There were like carrier proteins in the membrane. Remember those?
Mhm.
And so a molecule might come into the protein. Let's say this is a molecule coming into the protein. It might attach right there. And then how does this protein bring it in? How does this protein bring the molecule in?
ATP.
Well, ATP can come along. It can phosphorylate it, and that changes the shape of the protein. And it might make it open up to let it in. And then there's usually some trigger. Like imagine a little trigger down here. So when this thing comes in, it hits the trigger, and that knocks off the phosphate, and then it goes back to the shape it was before.
Whoa.
So as long as you got these on your membrane, it'll let in the molecule that's necessary. It's got a specific shape to bind to that molecule. And then the ATP has to do the work. So you have to create ATP, and so it requires energy to change the shape of that protein and let it go back.
Okay.
Now, it's not always used to let stuff in the cell. It could be used to change proteins in other ways. But phosphorylation is big. That's how the cell does stuff, often through phosphorylation. Whoops. Is this still this section?
Yeah, it is.
7.1? So,
Yeah.
this is called substrate level phosphorylation.
No.
So, here's the substrate attaching to the enzyme. And not only can ATP drop off a phosphate onto a protein, but we can take ADP and P and put them together using this process called substrate level phosphorylation. So here's ADP. Here's your substrate that has a phosphate on it. This could be a protein with a phosphate on it. It could be something else that has a phosphate on it. Maybe it's a lipid, a fat that has a phosphate hanging off of it. It can attach to this enzyme. The ADP attaches here. The substrate with the phosphate attaches here, and then the phosphate gets transferred to the ADP to make some product, and we've made ATP. We call that substrate level phosphorylation.
ADP before it gets liquid?
Yes. ADP stands for adenosine diphosphate, which means it has two phosphates. Di means two. So when it gets the extra phosphate, now it's called ATP, adenosine triphosphate. It has
Kind of like a train. Okay.
adenosine triphosphate. So here's an example. We see substrate phosphorylation in glycolysis, which we're going to start on tomorrow. This is the substrate. It's called PEP, phosphoenol pyruvate, which has a phosphate connected to it. That's why they call it phosphoenol pyruvate. And it connects to the enzyme, and ADP connects to the enzyme, and then we pass a phosphate to it. You know, if this ATP is going to be floating around doing stuff for cells, we have to make it somehow. And this is one of the ways it's made, substrate level phosphorylation. It's just fun to say. Can you say substrate level phosphorylation?
Substrate level phosphorylation.
Isn't that fun?
Very fun. Possibly.
So, we're going to study the mitochondrion, where a lot of the reactions to break down glucose occur. You got to know all the parts of the mitochondrion. Here's my big mitochondrion on the board. Here are the parts. It's got an inner membrane. It's got an outer membrane. It's got a space in between the two membranes. That's called the intermembrane space. Inter means between. The middle part of the mitochondrion is called the matrix. Like the movie, The Matrix. When they said, Neo, do you know what the matrix is? That's when I stood up in the theater and said, it's the middle of the mitochondrion. My favorite joke. I didn't really do that, but I thought it. I did think it. So this is the matrix. Matrix is just kind of a general word for a space. But anyway, that's the matrix, the inner membrane, the intermembrane space, the outer membrane. And do you notice that the inner membrane is folded? The inner membrane is highly folded. You can really see it here. It shows it well on the screen. See all the folds of the inner membrane? Can you guess why it's all folded like that?
For more surface area.
Increases the surface area. Very good, Cartier. It increases the surface area because the folds of the inner membrane contain proteins on it called the electron transport chain. Electron transport chain proteins. And I have shown an electron transport chain right here that's going to move electrons along and help make ATP. Well, I'm just showing one electron transport chain. There are electron transport chains all over this thing. And so the more folds you have, the more surface area there is to hold more electron transport chains. One mitochondrion might have, I don't even know how many, 1,000 electron transport chains on it. And remember, this is not a two-dimensional structure. It comes out at you. So this mitochondrion, if it were real, would come out like this, and it would go into the board. It would be this giant pill, and this membrane is folded all kinds of which ways.
3D.
Like the wave.
Like the wave.
Whoa, okay.
All right, Willis out.